So it's great pleasure to be here. So it's great pleasure to be in my country as well. Thank you so much, Dr. Hisham, for the invitation. And today I would like to speak about what is gene therapy, approaches and the strategies, and also what can be done beyond the gene therapy. Gene therapy speaks about the mutations, about the coding part of the genome. Anyone in this hall know what is the percentage of all genes in our genome? Anyone knows? It is 1%. Our genome has only 1% of genes that give us the proteins. There is 99% of our genome non-coding DNA. What does it mean? It means there is 99% can cause diseases. So today, in fact, I added some slides just now because I would like to highlight some things about my place and in general about scientific research. Since this symposium is organized by the scientific research section, scientific research is not about one discipline, it is about many disciplines. So that's why I would like to speak about my institute. I'm coming from Agora. So I'm in Faculty of Medicine, University of Geneva, but my lab and my team is located in Agora Institute. Agora Institute is in Lausanne. And that's how it looks like. We have a really amazing view, but the most amazing part is we are 6 institutions working in the same place. We are coming from EPFL, that is the Federal Institute of Switzerland, that's for technology mostly, and University of Geneva, University of Lausanne, Medical School of Geneva, Medical School of Lausanne, and Ludwig Cancer Institute. All these people are in the same place to do translational research, to do precision medicine, to do real research. That's why the research is not about one discipline. We are clinicians, we are scientists, we are computer scientists working together to solve a problem. So the research is about multidisciplinary. So what we do there, if you look about, we are working in 6 directions. All of them go in one topic, is about precision medicine. So what is precision medicine? It is all about genetics, about genome. How can edit our genome? How can use it for diagnostic? How can use it for therapy? How can use it for treatment response? That's what we call it precision medicine. I'm not a physician, but all doctors here can tell me, when I have like multiple patients with the same disease and give them the same medicine, they get the same outcome? I'm pretty sure no. That's why we need to look for precision medicine. And the precision medicine is mainly about genetics and the computer science. That's where I am working. That is the unit of precision medicine, in particular in precision oncology. And there is a link, by the way. What does it mean cancer? I'm working in cancer, yes, but what does it mean cancer? It is a cell lost, it is identity. And that is exactly what's happening in hearing loss. The cell lost identity. Any disease, any cell can lose the identity, it means it became in diseased stage. So what can apply for cancer? It can apply to any disease, lost the identity or became mutated. So that's just a very short introduction. What is gene therapy? It's just using the material of DNA as a pharmaceutical agent to prevent or cure disease. So instead of giving like drugs or medicine, we're giving short piece of DNA. That is what is briefly or simply what is gene therapy. So that's happening by insertion a piece of DNA to unhealthy cells or tissue. That is exactly what's happening. To insert that, we need a carrier. So we need something to carry this piece of DNA to this unhealthy cell or tissue. To first of all, to supplement mutant, so there is defect in this part of genome, and replace it with functional one. Someone can work. So this part has defected. We replace it with functional one. And we transfer that with vector that is mostly viral. So we carry this part of gene to virus and we can transfer it to the cell. And for historically, the first gene therapy, it was in the United States. It was in 1990 and was treated for ADA disease. So that is the first treatment of gene therapy. So there is 2 types of gene therapy. So that's based in our cell category. The first one is the germline gene therapy. That is, you can apply it for sperm, ovum, and the early embryo. And from there, we can modify the genome and or DNA. And this editing can continue for all generation and can affect all cells. In the opposite way, when apply this for somatic cells, you can edit a specific part of genome, but that's really restricted of this cell. And if it didn't go for the division, it means the gene therapy is really lost. And this one is not linked to generation, so it's only linked to this person. So I will speak about some points just to highlight the difference between these two types of gene therapy. About the germline, it's straightforward one. Just we take a piece of DNA and deliver it, and afterward that can be divided to the cells and go for generations. All cells have function, so because in somatics, in fact, not all cells have function because sometimes we go for off-target base of DNA. However, for germline gene therapy, all cells are functional, but there are a lot of concerns, because ethical concerns, because if you are working with sperms or ovum or embryo, that is really very big question about ethical approval. And I think most of us know about the story of a Chinese doctor who worked or modified the genome of baby, and he went now to the jail and they closed his lab. And I believe most of countries, they don't give really authority to be able to work in embryos. So the result is good or bad. It can be good or bad because we don't know what's happening in the embryo. However, in the second type, it's about somatic cells. It's tricky because we need really to go for a specific gene and replace it, switch it on, and that is not easy in our human genome. Because most of gene is not only about single gene, it is about multi-genes working together. So targeting only one gene can solve some problems for certain genes, but cannot solve all genetics situation. And that's very clear from the first trial in 1990. We have only few diseases are cured by gene therapy, because that can only targeted by single gene, and the rest of them can work as a multi-gene. And that's why I talk in the second part of my talk, how the whole genome organizing together to make these genes working in all directions. So I back to these issues of somatic gene therapy. It is very short life. Sometimes we lose it because the cells are not dividing. Also, that is, but there is some benefits of it. There is fund, there is labs, there is permission, there is regulation can allow people work in that in the opposite way of germline gene therapy. To do these two types, there is two methods. That's in vivo and ex vivo gene therapy. What does it mean in vivo? So that is we take the piece of DNA and put it in virus, and it has a viral infection that can throw that's one part of the in vivo, and second part non-viral to make capsulation with nanoparticle or liposomes and carry this part of gene and deliver it to the body. The second part is ex vivo. We take already sample from the patient and start to modify this gene editing this patient samples until we correct this gene, and afterward inject this cells after correction to the body. So that's the two ways when we can work in gene therapy in vivo and ex vivo. And this one is very famous in cancer, especially for CAR T cells therapy. That's one type of immunotherapy when we have modified the T cells to have antigens, and you go and you can kill cells inside the cancer, and it's very expensive ways. It can cost about 200,000–300,000 per patient. And so that is already has approval in blood. That's CAR T cells as ex vivo gene therapy. However, in vivo therapy, it is still also one of the examples that is blindness, who can inject directly to the eye and has a successful story using viral vectors to treat cancer, to treat blindness. Okay, thank you. Thank you. So now, since the most of in vivo gene therapy is about viral infection, how can do that in virus? So that is just a scheme how to summarize the types of vectors. That's viral and non-viral. The viral, this type of virus can carry the DNA to go to our blood. Retrovirus, adenovirus, adeno-associated viruses, and the herpes simplex virus. However, for non-viral vector, there's a carrier for the gene we are treating. There are physical methods and the chemical methods. Physical method like gene gun, so that's gun or gun, we can enter the gene for using this physical method, and the chemical method that's using different ways like nanoparticles or liposomes and so on. So to do this, to make the viral infection go to the body, we need to have editing techniques That's allow after the virus get into our body, we need to be sure there is a way to cut our DNA to replace it and pass this DNA to our genome. And that's, I believe, the most tricky part, because the precision of the cut, the precision of the busting or of the DNA, how can target specific gene. There is a lot of off target, because it doesn't go only for this part, it goes for different parts, and that's affect the gene regulation. So that's the most tricky part. And this part, in fact, has a lot of evaluation, started by different techniques as a gene editors. Let's see that scissors go and cut our DNA, and that is carrying this material to replace it in our body. So what is this scissors? The first one started with meganucleases. That's the first try when we have bulk of protein go for our DNA and stick for it. And the evolution of this editor named zinc finger nuclease, and the evolution of this one afterward. Okay, I will speak in details about each one, but these three types came as an evaluation from the meganucleases. And in 2011, it was selected by nature as the method of the year, because that is very important topic. When you go to our genome and cut and replace, so that was big advancement in the field to have such editors or such scissors can cut our DNA. Afterward, became in the era of CRISPR-Cas9. That's more precise. That is the most precise, I would say, is not scissor, it's tweezer. When you really pick up very precise part of the gene and start to edit it. And a few years later, it became selected by Science 2015 as the article of the year. It was the era of gene editing. And recently, a few or 3 years ago, it came by Nobel Prize. 2 people got Nobel Prize for this gene editing ways, especially in CRISPR-Cas9. But all of them, in fact, came from as evolution. No one makes everything from scratch. So that's kind of evolution, and the people building in the work of each other. I will speak. I would like to speak about each one and why it's important to look what is happening before to build in it to improve the precision, and that's very important for research. So the first one, or the first method as a scissor or as an editor, it was the meganucleases. Meganucleases that can recognize our DNA, and the cut long sequence is about, let's say, 40 base pair. That's a very large one. That's not very specific. And when you go for when you target a specific gene, it means now you are affecting with this long base pair other parts of the genome. But there is benefit for this became less toxic, because that is decrease the off-target genes affecting by this editing, and that is due to the high DNA sequence specificity. So that is why it's less toxic. That is to construct such plasmid. That is really takes a lot of time, and it's very cost a lot of time, and also takes a lot of money as well. The evolution that's applied to different disease models and in plants and bacteria and so on. But it didn't work for human genome as far as I know. The evolution of this editor, it was zinc finger nucleases. When it started to decrease the base pair or the target size, and also it started creating by creating double strand break for this part, and generate very precise genomic part you can cut and replace. So that's happened from 2 function domain. That's the domain for DNA for binding and the domain for cleavage. So one bind and another one cut. And that is really one of the first editor or scissor applied for application for gene therapy. That is zinc finger nucleases. That's a very important one, because people start using gene therapy afterward, and also for the effort for treatment HIV. In the same group, there is also TALAN. That is transcription activator like effector-based nuclease, and that is decrease the off-target effect comparing to zinc fingers, and the construction of DNA binding is easier. So people start evaluate decrease the specific binding, decrease the off-target domains, and also make it very precise. That's how can things move afterward. And also that has a lot of application in gene therapy and even in plant crops and so on. Now comes to CRISPR. That is the magic of gene editing. When you have really very precise or tweezer can go for high precision for cutting DNA, and that is can combine with case protein. That is a big protein who will go and replace and bring the viral or the part coming from viral and can make it for transcription. And that is the most precise one comparing to other techniques. But still, really, it has a lot of issues because many people would like to use it in embryo. There is a lot of it's difficult also. It's very easy to design, but it's difficult to maintain it in our body because it can lose at some point. But really, that is opens the door not only for treatment or gene therapy, for mechanistic study, because we know now you are targeting a specific gene or a specific part of the genome. That's how it looks like when you compare between the all categories. You can see the most visible one for application is CRISPR-Cas9. All of them making double-strand DNA break, and that is can also go for two mechanisms for the DNA break. That is homologous combination and also non-homologous and joining mechanism. So you have to cut the DNA. You have to bring a piece of DNA and make this connection afterward. I think I said all of the information between all of this comparison, but I want to see this one nucleotide pairing. So that is the most precise one. That's comparing for CRISPR-Cas9, and you can see here is 20 base pair, and it's 22 base pair, and that's the shortest comparing to other ones, and that is highly precise, and there is many application for each one. So if you like to have this comparison, I can share it with you. So what are the strategy? Now we know what we need, what kind of cells, what's the types, what is the ways, ex vivo or in vivo, what we need to get inside our body to make this editing. What is the strategies now? There is 4 strategies for gene therapy. The first one is the gene augmentation. I would say that's the most easy one. And targeted killing of specific cells. Although that is considered as a strategy for gene therapy, but that's the most important strategy for cell therapy. Targeted gene mutation correction and targeted inhibition of gene expression. And I put this one by end because really it's all about gene expression. If you have incorrect gene expression, we have all diseases. So the first one, it is about gene augmentation. We have cell now no function. We give them, we give this cell this gene with function, and from there it starts gene functional normally, and you can using all of these editors to make it. So that's the most simple one. Treat diseases caused by mutation. If you have muted one, straightforward, replace it with another functional gene, and this can continue and make functional afterward. That's the first strategy. The second strategy, I will say that's the most tricky one and the most elegant one as well, because it's combined between gene therapy and cell therapy. Gene therapy is not enough. I'm sorry to say that. Gene therapy is not enough. Gene therapy needs a lot of therapies around to make it work. So what is happening when we target killing of specific cells? That is very famous in our cancer field. That's, as I mentioned, about CAR T cells and immunotherapy. We have 2 ways to kill the cells. Either to carry gene to make cell suicide, and from there we trigger the apoptotic pathway, and from there we can kill the cell. The another way, the DNA, we mix the DNA to the cell carried with immune stimulating genes, and from these genes can make protein markers for this diseased cells, and from there now the immune system can recognize these cells and kill it. That is why it's very important, this technique in gene therapy and cell therapy. For cancer people, that is the most important one, because why our body cannot treat cancer? Because our immune system cannot see the cancer cells. That is very shortly. If immune cells can see cancer cells or recognize cancer cells as cancer cells, or any disease cells as a disease cells, or any cells loses identity, it can go ahead and kill it. And that's the idea of immunotherapy. So that's why we give the DNA immune stimulating genes, and from there we can tag the cells afterward here. And these genes can express markers, and this marker can recognize by immune cells. And these immune cells now can make it. So our immune system can work and kill these diseased cells. And that's why it's very expensive, because we need a lot of engineering to our genome, and you can edit it and afterward implement it to our body. The third category or third strategy is called targeted gene mutation correction. So in this case, we don't need to correct the whole gene. So that gene has just mutation in some part, and you can take like X gene here in diseased gene, let's say X gene, and just we correct this gene and became normal phenotype. Just correcting the mutant part of the gene, we do not replace the whole gene as well. And that's one of the benefits of CRISPR-Cas9, because the precision can go for only one base pair. So it can go for the diseased, let's say, muted part in the gene and correct it directly. The last one is about the gene expression. And by the way, that is the latest Nobel Prize, is about microRNA. How can block the gene expression afterward of cancer cells? That's just the latest Nobel Prize about it. So that is the targeted inhibition of gene expression. Gene expression, if it's corrupted, it means the cell became diseased and they start to stimulate other cells to become in disease stage. So the idea now to block this gene to make the expression. So that is the cell has a faulty gene, has default, and we need to block this one. And we block it during the RNA interference. We can use small molecules, like small RNA molecules, or short herbarium RNAs, just to silence the gene expression. So that the gene can express and they produce RNA to make the protein, but we can stop it at the mRNA part. That's the post-translational part. That's why I would like really to make it here. Do you think our genes making the gene expression? No. It's coming from we see the trash part of genome, the 99% of our genome. As I said, the coding part of our genome that makes all proteins, all genes, is only 1% of our genome. 99%, people consider it as a trash, but that's not reality. Recently, in fact, Dr. Said has mentioned about it, about the genome organization. That is the key part of our disease and how can our cell lost identity or became healthy or unhealthy. There is successful stories about gene therapy. So since 1990, there is a lot of trials, but just a few successful cases. Immune deficiency, that's the first one. Blindness, hemophilia, cancer recently, and this is the first approval for gene therapy, by the way, for blood diseases. That's we have a company in Switzerland, in fact, working in this one, and we can treat people nowadays. These are the successful stories only for all diseases. There is limitation, yes, for gene therapy. There is no way of treatment. One way can treat everything. It is a very advanced one, but it cannot cure everything. So the delivering is one of the limitations are technical challenges. The delivering of the gene to the right place and switch it on, that is not easy, because it's not on and off. It is regulatory elements around our genome. There is no single gene working alone. There is kind of synchronization with other parts of the gene with different enhancer and promoters. The immune response, sometimes our body doesn't like a piece of DNA coming from outside. So our immune reaction or in this part can reject this gene editing. And the virus as a vector affects the host. Still we give our body virus. All of us know about the situation about COVID and how can they develop this in a very fast way for viral or non-viral way. There is also issue about the funding. That's required a lot of regulatory bodies and systems to approve gene therapy, and at all scales. We need really governmental work, with institutional work, with like non-governmental also should continue in this support. So it's a lot of work. It's not decision of one, it's the decision of many organizations. It needs also collaboration of knowledge. It's not only one discipline. It's about cell biology, molecular biology, biophysics, imaging, physicians. It's not only one knowledge can enough to make this one. And the innovation in R&D needs also incentives. So you need to encourage people to work in that. So that is difficult to make it as a straightforward one. There is ethical barrier, because if any one doctor or anyone has this knowledge can make editing in our genome, so can make some kids heroes and there are kids are really stupid. So that is, we should be barriers for that to stop. And also the companies, especially cosmetic companies. Cosmetic companies, they have a lot of money, and they work in gene editing and therapy, and they can take the patent of that. They can block this therapy for themselves, and we have to pay them a lot of money. So there is a lot of concerns, ethically and legally concerns about it. And I believe the cost, CAR-T cells is $300,000 per patient, and the success rate is about 50%. It's still so expensive. So also, who can decide this trial is correct or not? It depends from one to one, it's depends from country to country. So it's not really a standard protocol for that. That is making gene therapy also has some success examples, but still has a lot of limitation. We need to work on it. I personally believe the limitation is always about 1%. We are speaking about that is okay. That is there is nice, beautiful image here about our genome, but I can, because it's dark, I cannot see it. But I can tell you, our DNA is 2 meters, as Dr. Said mentioned, and we have in our body about 20,000 protein coding genes. So we have this coding genes 20,000, and this 20,000 is only 1 to 2%. There is 99% rest of our genome. We don't know anything about them. So it's not fair to work only in 1%. We have to work in the rest of our genome. That's what we're doing in our institute and also in my team. And that's the challenge. If you look about this 2 meters of DNA, to make it fit a nucleus of 5 to 10 micrometer, do you know how many fold we need to have in this one? 1,000 fold. Because to make 2 meters and 5 micrometer, we need really to be folded and organize it in very sharp way, in very systematic way, in very elegant way, to be accessible to protein, to start transcription and gene expression and so on. Any shift in this organization, so starting in for compartment level, to specific domains, to loops, to that is the gene when that trigger oncogene or not. Any shift of this organization, that can make cancer. Because instead of to make repression for gene here, can make oncogene here. So any small shift can make not only cancer, it can make any disease. And there is no mutation here, by the way. So even when we speak about sequencing, it's great to have sequencing. Sequencing is not enough. Sequencing speak about only 1%. But that 99% is about organization. So genome organization, I highlighted in very bold because that is extremely important, really. Genome organization and epigenetic modification have reversible change in gene expression that don't alert DNA sequencing. So any change in the organization and gene expression is not linked to the sequence. So what happened? We have million cancer, many type of cancer is not related to mutation. We have many diseases are not related to mutation. It's really related to the genome organization and epigenetic modification. And that's the way, and that is how it works in chromatin or the DNA. That's the chromatin is the backed form of our DNA when histone comes to back this 2 meters to fit in this 5 micrometer. On top of this chromatin, we have epigenetic mechanisms helping to function in this DNA. That's DNA methylation, histone modification, remodelers, and non-coding RNAs. I just give you some examples. When we have DNA methylation, we're adding methyl group to our DNA, make it unfunction. So it became accessible and can start another gene expression program. So that is, although it's very important in development for embryo, but in somatic cells, it is really dangerous, can cause cancers. Histone modification, we have DNA methylation here also that can make cancer. Recruiting wrong remodelers to our DNA, that's opening DNA or the compact, it's all about compaction of our DNA. Opening this compaction can a wrong place or with compaction level is not necessary, can make cancer or any disease. Again, cancer is it's not a disease, it is a changing of the cell state or cell type. That is a cell loses the identity as any disease. And non-coding RNAs. So it's all about chromatin and epigenetic modification. That represents the 99% of our genome. That's what I do in my team. I'm really in love or really full and big fan with the senescence. What is senescence? When we became old, still our genome is the same, our cells function, but that is a big marker for our age. So that is a golden marker for aging. That is, we can find a lot of senescent cells in our body. What's happening in this senescence? Our genome totally reorganized. There is mess of organization of our chromatin. That is the genome, and this organization messed up and make some blobs, and this blobs is only about chromatin and epigenetics. But it's only coming by aging? No, it's not coming only by aging. When we treat a patient, making them chemotherapy that makes cells become, and there is famous statement here in age, we say he looks like more older than his age because he has more senescence, by the way. Because his cells stop to divide, his cells became fatigued and start secrete bad materials or harmful chemicals. We call it SASAB, this colored dot. The secretion of this SASAB can make cells stop. First of all, it can stop immune system to work. Second, it can trigger to senescence. So if you have only one senescent cells, it can make many senescent cells. And it's not really linked to one cell type. In all cell types, we can have senescence. We can senescence of skin, senescence of bladder, we can senescence of eye. So that is how it makes senescence is not only about aging. That is the one part, the replication stress. What does mean replication stress? When our cell divide over time and stop, became fatigued. And that's why we became old. What happened? Is that the only reason to become old? No. Oncogene activation. People with cancer, when their genes are activated, they make senescence. They introduce senescence in our body. Oxidative stress, the pollution around us, fast foods, that make also as oxidative stress. So unhealthy food make you look older than your age. And any damage for DNA, any exposure for any chemical exposure for radiation, for anything can make DNA damage, can make more senescence. And of course, that chemotherapy can make senescence. So there is not only about related to age. Senescence is not related about age only, it's related also about our daily life. What is pollution around us, oxidative stress, what treatment we have, and so on. So the question I'm asking to myself and my team and my research, can we make all younger if we get older here? Can reverse our age? Can treat cancer with that? Yes, we can. Short answer. I will go in details now. So, as I said, there is two categories. Let's say that is the one come by nature. When you have replicative stress, that's coming by age. And another way can change the gene expression, make genes are oncogene. When you had a look, that is one of our papers, the organization of genome here, that is how it looks like as a healthy cells. The genome is really organized, part of chromatin named lamina associated domains start to get inside the nucleus and they form entity, this blob, that is mostly about the methylation, how this methylation happened in our chromatin. What about another type when we became older? That's the replicative senescence. When we make this, because we cannot make experiment on body for 60 years to monitor that, we found the drug that can make cells become senescence and make this in cell culture and came with remodelers. And you can see this remodelers has the same, by eye, when you look to the microscope, you have the same entity of chromatin blobs, but with different structures. So the drug, but by end, both of them are senescence. When we treat both of them, one of them will have effect and another will no, because both are different. This remodelers and this methylation. So we cannot treat both of them in the same way. That is the beauty of working in the whole genome, and you can see it by eye and go for molecular details to distinguish between the cell states, different cell states for the same cell type. So now we're speaking about the cellular level, but our body is not at cell level. It is about tissue levels. Here it looks like our tissue. We have the cell types, we have stem cells, we have fibroblasts, we have immune cells, and we have triggers. Any of these causes or any of these things can cause senescence. We can trigger any cell type to senescence. the problem is not having senescence. Senescence trigger another cells to become as a secondary senescence. So that is became as a source of harming for our body, because the secretion of SASAB or this harmful chemicals can affect the neighborhood of the cells. So that can trigger immune cells to become senescent immune cells, or senescent fibroblast, or senescent whatever cell type inside this tissue. So what does it mean? That's for cancer treatment, and I can link it now to hearing loss. Believe me, I will link it to hearing loss now. The problem with senescence, it has a dual effect, paradoxical effect. It is very important to our body, and at the same time, it can cause cancer and it can treat cancer. So for tumor suppression, a little of SASAB expression that is can trigger our immune system, so immune system start to work and it can kill cancer cells. That is the most important part, so it can stop cancer. When senescence, as defined in early time, is defined as a mechanism to stop cancer. But however, when it has this senescence secretion in very high amount, it can trigger many harmful things. It can even cause tumor, because a lot of inflammation happening from this one. This inflammation can stop immune cells, it can trigger tissue remodeling, it can make vascularization, it can make many things, stop the cells, the tissue from functioning. So now we don't know when use senescence in good way, when we use it as a bad way. We cannot target it in our body easily, because they have different cell states, because our body can has, our air can have senescence, our eye can have senescence, so we don't know which type we can target now. So we are working in that. We have a combination between different therapies. But if you look here, how these cells, now the cell, that's normal cell, became aging. Can we eliminate senescent cells? Yes. There is new kind of treatment, we call it senolytics. The senolytics, that is kind of drugs, can go and specific go for senescence and kill them and eliminate senescence. And when we really, people apply this one in the skin, and very old skin became very young one. So that can trigger back, or eliminating this senescent cells can make our body again back to normal. But the idea now, why we have to wait until we have senescence? Can we stop it in this stage? Yeah, there is some trials. People working in internal medicine know about metformin for diabetics. In fact, last year it took the, so the article came out in Cell, and using metformin as a AMPK activator, so that's blocking the pathway from here, and they found metformin can really reverse the age of rats. But that is, I'm not advising anyone to take in metformin, but that is the idea. When you are blocking the pathways, that is the pathways of senescence. When you are blocking this one, you start to have stopping senescence to go for a mature state. So we are blocking it from the way. So can reverse it? I think we can at some point. Can reverse, we become more younger. I think women here will be very happy, because we become always younger here. So that is the effort to stop senescence at the beginning, and already we have two D and C, that's kerosene and cysteine. That is can really from now, the combination between them can eliminate senescence, and they can that's help for cancer curing, and also can help for other diseases. Can you use the same cocktail for hearing loss? Yes. Recently, just a month ago, this article came out, is about the senolytic treatment, alleviates cochlear senescence and delays age-related hearing loss. So the combination of this, what we give to cancer, they give it to the rats, and that was organ of Corti, and they see that is can really delay the hearing loss, because we are eliminating this senescence cells in the air. So now I will speak just one slide about my research, and that's in cancer. I wanted to show this one, as I showed, that is senolytics, that we use it in eliminating or killing senescence in cancer, is worked out for hearing loss. Can you use this approach, the most recent approach in cancer research and treatment for other? It could be, but we need to try. So what we do, I'm really very lucky in my institute, because my institute is translational. We have a bridge in our institute to the pathology department, when we can get fresh samples and work immediately on that. So we have our patient take tissue biopsy, and instead of to have it in, because to have a drug screening or drug selection, it is not effective to work in just cell lines. We need to work in real tissues. There is new techniques named organoid. When you take a biopsy from the patient, let's say from the liver, and mix this liver in a tissue in the lab. So that is really represent the tissue of the patient, because we can culture this biopsy in 3D. It has all cells. It's exactly like the organ of the patient, but in our lab. We are taking this biopsy, making tumor microenvironment. So that means we have these cells, let's say cancer cells, and the microenvironment like immune cells and the senescent and the others. We really mimic the organ of the patient in our lab. So what we call it patient-derivative chromatin organoids or tumoroids. Now we have something as a real, something tissue and in 3D. The most important part here, I would believe, to see what is happening in our genome in 3D. So that's why what I do in my team also, we develop 3D single-cell imaging techniques, not only for chromatin, but also for special proteomics, the protein inside, and also for transcriptomics, also to see the transcript messenger RNAs. And that's how it looks like our organoid. We have the cancer cells, immune cells, and you can from here have markers for DNA and protein, whatever protein we are looking for. And from here we have single-cell analysis. We can know now differentiate between patient to patient because we have the cellular level. And from there, we can see the cell-cell interaction. Cell-cell interaction, one of the most markers now to treat and cure cancer. And from there, we can have markers based on chromatin and RNA from this biopsy for each patient. That's why we call it precision medicine, because we know that for a specific patient, he has this information. So what we can do afterward, study the heterogeneity of this patient, how the cells are heterogeneous, and how can assess the combination of therapies. So, since my lab really interested in senescence, we target senescence inhibitors and epigenetic therapy. There is epigenetic therapy just work in the epigenome. That's not related to mutation at all. And the senescence is not related to mutation as well. And even immunotherapy is not related to mutation. And also there is other targeted inhibitors that work in pathways. So all therapies we are using here are not really related to mutation. We don't need to correct it. Having all of this information, we can really know what is the molecular pathway of each patient. We can link the SASAB to immune evasion and the therapy resistance. That is why each patient has different outcome, because there is resistance for our body. So we can now define the resistance of the therapy. We can link the molecular pathway to each therapy class. So this one, let's see, this patient will take combination between this 1, 2, but the second patient will take combination between 2 and 3. Because everyone is different and everyone has different genetic materials. We are working only in solid tumors. No, we're working also in blood. We are taking blood because blood also has immune systems, can have microenvironments, so we can have this treatment also, not only biopsy from our tissues, but also from blood. I'm sorry. So what I wanted to say, that's what we do in cancer. I show you in cancer also, we found the combination, it is treating the hearing loss. I think such a strategy for research also can help to treat other, as cancer is losing of cell identity. The hearing loss also the hair cells are already lost their identity. And by end, I would like to thank you so much for your attendance. And that is this picture taken by myself. That is in Geneva. And I would like to thank all people support. The funding agency give me the money to do my research. And thank you so much.